Hello dear colleagues, I'm Zoribel Sosa from Dominican Republic, and in this video I would like to share with you my learnings from this week. We are currently talking about study sample and what do we need to know here. First, we have to be clear with 4 words that are population, target population, study population, and sample. Population, people being there, living, being happy. Then we have target population, who is the group that the investigator is interested in knowing and have conclusion. After target population, we have the study population, and this group has to be very specified. Has to be accessible, has to be agreement with inclusion and exclusion criteria, and has to be representative of the whole target. After the study population or accessible population, we have the sample. Sometimes the sample and the study population could be the same, but in another case, not. In another case, the study population is higher than the sample. Now that we are clear about study population and sample, we have two challenges. The first one is defining the study population, and the second one is sampling. And what is important? What we need to know? What we need to consider in the meantime to choose our population? First, defining our population, we have to be clear with two things. Number one, we have to be very specific. For example, as the book says, if we are going to conduct a study in patient with a stroke, we only not can say inclusion criteria are patient with a stroke. No, we have to be more specific. And in the other hands, we have a problem if we specify too many things. In this case, we couldn't be able to reach some patient for the study. So we have to be moderate, having criteria and exclusion criteria according we are interested in investigate. Defining our population, we have to be clear with two things. One is internal and another is external validity. And what does that mean? Internal, it is how accurate the outcomes and the conclusion drawn from the findings that we are representing at the beginning. And external mean how applicable are our conclusion in the study. And in internal validity, we have to be precautive with the bias and try to minimize those risks in our area. Our second challenge is sampling, and as doctors frequently says on the book, the more representative the sample is, the more generalizable our study will be. So we have to be careful. And in this topic, we have two types to choose our population. One, it is probability sampling, and in the other hand, we have non-probability sampling. And what is this? In the side of probability sampling, we have sample random sampling, systematic sampling, stratified sampling, cluster sampling, and disproportional sampling. I would like to result the one who is cluster sampling. While I was reading the book, I was interesting. I can choose a group of people with characteristic, and this could be a cluster sampling. One way to do the sampling being a probability is the sample random sampling. This is like the ideal, the best one, the one who has minimal risk, but the way to make this is more expensive. We have to invest more time. On the other hand, we have the non-probability sampling, which we can find convenience, consecutive, and another one is snowball sampling. I have experience in this. Now that we mentioned some type of sampling to choose our population, I would like to share my experience. I work in a primary care center where we do phase 3 clinical trials, and we choose the population by convenience. For example, we work with people living with HIV. Our patients come to the visit, we told to them to the current study, criteria inclusion, exclusion criteria, what about the study is, why the study is important for them. We talk about the informed consent if the patient is interested, and then we start enrolling in the study. So we make our sampling only by convenience. In other hands, I had another experience last year with my colleague Laura Santana, who is a teacher assistant at this. We made this study last year about acceptance and confidence in the COVID-19 vaccines in focused clients who attend the Dominican Institute of Virological Studies. What do we do? Well, we have two arms. One, 100 people living with HIV. In another hand, 100 people HIV negative. We were approaching to all the clients who came to the center in the space where we are recording our data. We talked to the study, we made with them informed consent, and if they accept, we enroll in the study. So my experience is only with no probability sampling, and I will love to work in a study with sample random. I would like to have this experience. I would love to finish my video giving my 6 recommendations in the case study. First, I will definitely apply the condition that simulate rheumatoid arthritis, since there is a very high probability of reducing bias, and it will diminish the credibility of the results from the study. Second, in this case, I will exclude polymedicated patients. In the sample, choosing the samples, I would like to be considered to have anatomal pathology results. I know it will be considered like he has a amount of money from the grant and he has to invest. But I would prefer to have a homogeneous target, and so we can have better results. For talking about recruitment, I think he will have some conversations with another colleagues who could refer patients from here from the studies. I think all people can be participant in the recruitment part. And for others, I think it will be interesting to give a close follow to the patients, calling, testing, saying your appointment is next week, how can I help you to be on time, and in this. And I share this based on my experience working as a data entry assistant in a clinical trial in Dominican Republic. It has been a pleasure to share with you my experience and my learnings from this week.